Assalamu alaikum, my dear students. So in our previous video, we had discussed about the different parts of microscope. I hope you find that video interesting. And if you're finding my videos interesting, please like and subscribe my channel. And let's move forward. As you know, the different parts of the microscope, the next learning outcome is to, uh, examine the different type of animal cells and the plant cells under the microscope using suitable locally available materials. Let's see. Let's start with the onion cells. But before starting with the onion cell, let's talk about the basics of the temporary staining techniques. What is the procedure of temporary staining technique? For temporary staining, you need a glass slide. A glass slide should be very clean, very clean. It is better to use a new glass slide. And if you find it in any kind of dust particles attached to it, the first step is to clean the glass slide. Otherwise, all the things would be visible in your microscope, and you will not be able to see your specimen clearly. So what is in a specimen? The specimen is the object you want to study. So for example, if you're looking at animal cell, the animal cell is your specimen. If you're looking at the plant cell is your specimen. So specimen is something that you are actually focusing on in your microscope. So after taking a clean slide, what you have to do is you have to place a drop of sterile water. Again, we don't want the water to be filled with bacteria and other type of microscopic organisms. So it should be a sterile water. It should be sterile and it should be clean so that there is nothing else which is hindering your observations. Okay, then you have to place your specimen, whatever specimen you are using. Like in the video ahead, I'll show you that how onion and human cheek cells are being used. So you will be you'll be placing your specimen. Then you have to place a piece of cover slip. A piece of cover slip is a transparent piece of glass. It's a very fine layer of glass, and the purpose of cover slip is to keeping your specimen, covered, protected, and also we don't want the specimen to drop away from drop off from the slide. So that is why we place a cover slip just to protect it. So how are we going to place the cover slip? If we'll drop the cover slip horizontally on the surface of the specimen, you will get a number of bubbles in it. So what you have to do, you can see that, I'm sorry, you can see that the cover slip is being placed on at an angle. It is better to place the cover slip with the help of a forceps or a needle. Place a needle under it, and then place the cover slip and just drop it on the specimen. So when it gradually drops, it removes all the air bubbles that might be trapped with, in case agar aap usko horizontally place karte hain. So it will remove all the air bubbles that might be trapped here. Once your glass slide is ready, just wipe out the excess liquid, because while placing the cover slip, some of the liquid leaks out. So what you have to do is, using a tissue paper, you have to wipe out the excess liquid, and your slide is now ready. Now place this slide on the microscope stage and observe it. Okay, you can use low and high magnification objective lenses in order to see your image. Okay, I hope this is clear. So this is the basic mechanism. Let's look at the two type of specimens, the onion and the human cheek cells. Firstly, let's talk about the onion slice. How are we going to observe the plant cells? Okay, for that, we are going to use the leaves of the onion. You know, the layers in the onion are known as the leaves of the onion. What you have to do is you have to cut the onion into two halves, and then you have to take one of the leaf, take a small portion, a very small cut of the onion. You must have seen that on the surface of the onion, on the inner side of the onion, there is a slimy, slippery layer that usually comes off very easily. You just, with the help of a forceps, you just have to separate this layer. And you have to place this layer on the microscopic slide. Again, the slide is supposed to be clean. Once you place it, it is better to use, you can directly use water, or you can also use a dye for it. Say, for example, let's use iodine solution. We usually use iodine solution for onion slides. So place a few drops of iodine solution on the specimen. This will give the color to the different parts of the cell and make it very easy for us to, visualize and understand. So you'd have to drop a few drops of iodine solution. Then again, as I've told you, the mechanism of dropping the cover slip, you have to place the cover slip at an angle and then just drop it. Once it is dropped, it will remove all the air bubbles and it will be covered. So wipe out all the excess liquid that is present and observe it under high power objective lenses. So what will be your observation? This diagram, as you can see, this is the diagram. This is the original specimen, okay? The diagram of the original specimen, a picture of the original specimen. Here you can see the cells and you can see the nucleus and cytoplasm and cell wall and cell membrane are very clear. So how, under the microscope, you will see that the cells are continuous. There is no gap in between the cells. The cells are usually rectangular shaped structure or cylindrical structures. And you can see it's very much clear. The cell wall and the cell membranes are clear, and the nucleus is very much visible. The other parts like the Golgi bodies or endoplasmic reticulum or mitochondria would not be visible under this magnification. For that, you need to use more advanced forms of microscope, but these structures are very much visible, so you can see that. The structures you can see and you can observe. What you have to do is you have to draw your observations on the piece of paper, and that's it. That's it for the onion slide. Okay, so let's move forward. If you have any questions, if you have any questions, you can ask. Okay, so as we have seen the onion slide, let's talk about the human cheek cells. For making a slide from the human body, you have to take a few precautions. For human cheek cell, we usually use a toothpick or a small wooden rod, or we can also use the cotton bud. The important thing is that we have to use, obviously, the sterile material. Why? Because why it is important is to keep things sterile, because we have to prevent ourselves from any kind of infections. So it's a very easy way of making the slide. You just have to scratch the inner lining, gently scratch the inner lining of your cheek, and a number of cells get attached with this stick. You just have to drop the stick, or you have to rotate the stick on the slide. So gradually all the cells will shed off on the surface. You don't need to stain it, or you can stain it, but using a small drop of water would also be sufficient. Again, cover it with the cover slip and observe it under microscope. As it is an animal cell, you will see a slightly different, a very different structure. You have seen that in the plant cell, the nucleus was on the edge, but in the animal cell, you will see the nucleus in the center. The plant cells were properly shaped, rectangular or cylindrical shaped cells, and the animal cells are usually irregularly shaped. They do not follow a exact shape, a particular shape, because there is no cell wall in the animal cells, and you would not see any cell wall again because there is no cell wall. The cheek cells are not all attached in a regular pattern as you have seen in the onion epidermal cells. The onion layer was all the cells were closely packed, but here this is not the case. You will see the bunch of cells, 3, 4, 6, 8 cells maximum, not more than that. So, or you might also see a single cell separated from the other cells. So this is all about, how to make the human cheek cells done. As we have discussed the precautions, what are the precautions? The precautions include are that you have to use the clean cover slip and microscopic slides. Cleaning is not a difficult task. You can use water, alcohol, or you can just wipe it with the tissue. That's it. The cover slips, this is an important point. The cover slips are very thin and fragile. They can be broken down easily, and they have very sharp edges. So while handling with the cover slip, it is important that you have to use the cover slip with the forceps. Avoid direct touch of the cover slip, and obviously the touching also affects the texture, because your fingerprints get onto the cover slip. That also causes a difficulty in visualizing your image. And more importantly, the cover slip might break. so it can cause some kind of small, cut on your skin. So to avoid that, it is better to use the forceps. One thing which is not mentioned here is the sterile conditions. It is very important to maintain the sterile conditions while handling with the animal plant tissues. Particularly when you are using any kind of animal tissues, it is very important to maintain the sterile conditions. You don't, while making human cheek cells, you don't need to scratch very firmly. It should be a very gentle scratch, so that so that it does not harm the inner lining of your cheeks. So I hope the process is clear. And if you have any questions, you might ask you're most welcome to ask in the comment section, and try to answer all the questions. I hope you're liking my videos, and I expect you all to, make your own notes, so that you can refer to my videos again and again, but still you need to make your own notes so that you are able to write down the answers appropriately. Like as we'll go further, inshallah, I will be showing you different type of past paper questions that are coming along. As we'll cover the topic, okay, what kind of questions can be asked in the exam and how you are going to answer them. So keep watching, and please subscribe my channel. Allah Hafiz.